Digestion of lipids. The major dietary lipids are triacyl, glycerol, cholesterol, and phospholipid. The average normal Indian diet contains about 20 to 30 gram of lipids per day. Western diet contains more than 2 to 3 times this quantity. Digestion in stomach. The lingual lipase from mouth enters stomach along with the food. It has optimum pH of 2.5 to 5. The enzyme therefore continues to be active in the stomach. It acts on short chain triglycerides SCT present in milk, butter, and ghee. The action of lingual lipase is observed to be more significant in newborn infants. Gastric lipids is acid stable with an optimum pH 5.4. Up to 30% of digestion of triglycerides occur in stomach. Digestion in intestine. The emulsification is a prerequisite for digestion of lipids. The lipids are dispersed into smaller droplets. Surface tension is reduced and the surface area is increased. The process is favored by bile juice or bile salt. Peristaltic phospholipids, bile salt, is important for digestion. It reduces surface tension, lipolytic enzyme in intestine. The pancreatic lipase with colipase and the phospholipase A2 and the cholesterol enterase. Digestion of triglycerides. The pancreatic lipase can easily hydrolyze the fatty acid electrified. This is the reaction of beta oxidation of fatty acid, which is important to remember that the first step is FAD dependent and the third step is NAD dependent. The acetyl coenzyme and the fatty acyl coenzyme, it is formed from the fatty acid which is sent to the TCA cycle, and further the fatty acid coenzyme further cycles. Summary of beta oxidation of palmitic acid. The C16, that is the 16 carbon acetyl coenzyme peptide. When the acetyl coenzyme, it is removed from the C16 carbon, the C14 is formed, and the C12 the C12, the 12 carbon atoms. From the 12 carbon atoms, the acetyl coenzyme, it is 2 carbon atom is removed and the further reaction formed is 10 carbon atom is remained. Further, this process actively performs till the single acetyl coenzyme, that is 2 carbon atoms are removed or are remained in the last. There's 7 cycles. It is the reaction for the triacylglycerol synthesis. It is synthesized from dehydroxyacetone phosphate. The common enzyme used is acyl transferase. The cycle starts from the dihydroxyacetone phosphate to lysophospholipids. Lysophospholidic acid, when acts with acyl transferase, the phosphatidic acid is formed. With the help of phosphatic phosphatase, the 1,2 diacylglycerol is formed. With the help of acyl transferase, the 1, diacylglycerol turns to triacylglycerol. There are two important fatty acids caused by liver adipose tissue and the fatty liver. The adipose tissue serves as a storage site for excess calories ingested. The triglycerides stored in the adipose tissue are not inert. They undergo a daily turnover with new glycerol molecules during the synthesis and the definite fraction being broken down. The adipose tissue normally the glucose and insulin level are increased. Insulin increases the activity of key glycolytic enzymes as well as the pyruvate dehydrogenase acetyl coenzyme. The carboxylase and the glycerols phosphate acyl transferase, the stimulate effect of insulin on the uptake of glucose by adipose tissue on the glycolysis and utilization of glucose. Insulin also causes inhibition of hormone sensitive lipids and so lipolytic is decreased. Adipose tissue in fasting condition are can be shown as the metabolic pattern totally changes. The protein from adipose tissue is mobilized under the effect of the hormone glycogen and epinephrine of the hormone. The cyclic AMP mediated activates the cascade, enhance the intracellular hormone sensitivity lipids. The phosphorylated from the enzyme is active with which acts on tag and liberates fatty acids. Adipose tissue and diabetes mellitus. The lipolytic is enhanced is noticed in diabetes mellitus. The insulin acts through receptors on cell surface of adipocytes. Therefore, the receptors are decreased, leading to insulin insensitive in diabetes. Adipose tissue and obesity. The fat content of adipose tissue can increase to unlimited amount depending on the unit of excess calories taken in. This leads to the obesity. Plasma insulin level is high, but insulin receptor are decreased. Some causes of fatty liver can be the excess mobilization of fat, toxic injury to fat, the alcoholism, calorie intake, metabolism of ketone bodies. Or can be the carbohydrates are initial for the metabolism of fat or the fat burn under the fire of carbs. The acetyl coenzyme formed from the fatty acid can enter and gets oxidized in TCA cycle only when the carbs are available during the starvation and diabetes mellitus. The ketogenesis is the ectoacetate is the primary ketone body while beta hydroxybutyrate and the acetone are the secondary keto bodies. They are synthesized axially from the mitochondria. The process are condensation, the for production of HMG coenzyme, beta hydroxy beta methyl glutari coenzyme. The third is lysis, fourth is reduction, and the fifth is spontaneous decarboxylase. The first step is the acetyl coenzyme joins with the second atom of acetyl coenzyme, that is the condensation takes place. Then the coenzyme SH given out for the formation of acetyl coenzyme. Then the second step, the acetyl coenzymes forms the HMG coenzyme. And the third step, the lysis takes place for the formation of acetoacetate and acetyl coenzyme. The acetone is formed in the one side and the second side the beta hydroxybutyrate is formed.